Okay, so if you were asked to design a molecule which could act as the genetic material in living things, where would you start? One of the features of the genetic molecule would have to be the ability to carry instructions, a sort of blueprint for the construction and behavior of cells and the way in which they grow together to form a complete living organism. Another would be the ability to be copied perfectly over and over again so that whenever the nucleus of a cell divides, it can pass on the exact copy of its genetic molecule to the nuclei of each of its daughter cells. We know what is a daughter cells. The cell formed by either by meiosis or mitosis from the original cell is called the original cell's daughter cells. Now there is nothing to make you understand but a genetic material, we know it must carry information and it must have the ability to be copied. It's normal common sense. Now, the structure of DNA and RNA. In organisms, living organisms, DNA and RNA is the genetic material. Now we need to know what is DNA and what is RNA. DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. DNA is the main genetic material, but ribonucleic acid is also essential. We will understand that in the interaction between DNA and RNA in details in the chapter of protein synthesis. Now, why do DNA and RNA have different names? That's because DNA contains deoxyribonucleic acid and RNA contains ribonucleic acid. We will see the difference right now. DNA stands for deoxyribonucleic acid and RNA stands for ribonucleic acid. As we saw in chapter 2, nucleic acids such as DNA and RNA like proteins and polysaccharides are macromolecules. They are also polymers made up of many similar smaller molecules joined into a long chain. The smaller molecules from which DNA and RNA molecules are made are called nucleotides. DNA and RNA are therefore polynucleotides. They are often referred to simply as nucleic acids. Okay, so now we know that DNA is a polymer, DNA and RNA are polymers, there should be monomers and both of them, the monomers for both are called nucleotides. So if nucleotide is the monomer for both DNA and RNA polymer, so why do they have different names? That's because DNA contains deoxyribose sugar, while RNA contains ribose sugar. Both are pentose sugar, but deoxyribose do not contain one oxygen atom which the ribose sugar contain. Okay, so we will see them right now in details. Now nucleotides. Nucleotides are made up of three smaller components. They are a nitrogen containing base, a pentose sugar, phosphate group. So pentose sugar for DNA is deoxyribose and the pentose sugar for RNA is ribose. So why it is a pentose sugar? That's because this sugar molecule contains 5 carbon atoms. Okay, now let's see these structures in more details. There are 5 different nitrogen containing bases found in DNA and RNA. In DNA molecules there are 4: adenine, thymine, guanine and cytosine, and RNA molecule also contain 4 bases but never the base uracil. These bases are often referred to by their first letter A, T, C, G, U. So first of all, we have known one difference between RNA and DNA is that the difference in the pentose sugar. Now we know another difference, as that is the RNA contains the base uracil while the DNA contains the base thymine instead of uracil. Now for the bases, we do not figure 6.3, we do not need to learn by heart the structure, the diagram completely, but we need to know that purines, bases are divided into 2 parts, purines and pyrimidines. Purines are adenine and guanine, while pyrimidines are thymine, uracil and cytosine. And that purines contain double ring structure while pyrimidine contains single ring structure. Okay. The pentose sugar can be either ribose or deoxyribose. As their name suggests, deoxyribose is almost the same as ribose except that it has one fewer oxygen atom. Okay, so let me show you the structure of the sugar. Let me show you the structure of the sugar, ribose, pentose sugar. it contains 5 carbon atoms. 1, 2, 3, 4. This is an oxygen, and number 5. We need to know the numbering of the carbon atoms. This is number 1, number 2, number 3, number 4, and number 5. CH2OH. Okay, so there is hydroxide, hydrogen, hydrogen. There is the base there. Okay, let's draw it later. Let's draw only the sugar. We have here hydrogen, hydrogen. Okay, and then here also we have hydroxide, hydrogen. Now this is the pentose sugar, but the difference between DNA and RNA, that is deoxyribose and ribose, is that ribose sugar contains hydroxide in the second carbon. This is ribose sugar. Now deoxyribose has this oxygen missing, and that is why it is called deoxyribose. And the oxygen and the oxygen difference between ribose and deoxyribose is in carbon number 2. And we will also know that in carbon number 1 the base is attached. Okay, so where is the duster? Now, the structure of the DNA or RNA is like this. Pentose sugar, phosphate, pentose sugar, phosphate. The base is in carbon number 1. This is the nitrogenous base, nitrogenous base, nitrogenous base. Now, one thing you may be confused. If you are thinking that this is the skeletal structure, then you are wrong. Well, that is because this is not a carbon, this is a oxygen. But for the simplicity, we draw it like this. So this is not a skeletal structure. In skeletal structure, this was supposed to be a carbon, but this is not a carbon. This is an oxygen. There is an oxygen. I just showed you before. And also there is one carbon here. So this is not the skeletal structure. So this is the monomer nucleotide. Okay, so this is the monomer, that is nucleotide. Okay, fine. Let's move on. Now, you may have one question. How did the phosphate ion came into action? Well, this is not in your book, but you should know. Since you are a student of biology and you took your higher biology, so the phosphate ion came from phosphoric acid, H3PO4. The structure of phosphoric acid is like this. Now, when phosphoric acid dissociate, Since it's an acid, it will dissociate in water to give up a hydrogen ion. It becomes negative. Okay. Now, after that, so this is like mono hydrogen phosphate. I'm sorry, trihydrogen phosphate. So you do not need to name it like that, but since this is not your chemistry course, so how do this ion, I mean like this trihydrogen phosphate ion, form a bond with the carbon number 5 of the pentose sugar? Okay. Now let's draw the structure of the pentose sugar again. This is the pentose sugar. This is CH2OH, and here is our trihydrogen phosphate ion. Here is an oxygen. Here is hydroxide. Here is hydroxide. This is an alcohol group. And this is an acid group. So they can form ester bond. From here, the hydrogen bond will be removed, atom will be removed, and from a hydroxide will be removed. Thus, a water molecule will be removed, and they will form a bond. That is the ester bond. So, how is the ester bond? After forming the ester bond, what will the scenario be? The scenario will be something like this. Say this is carbon, 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 oxygen, carbon, CH2, O, P, there was another O, there was one oxygen negative, and there was another hydroxide. So, and this is the carbon number 1, so there will be a nitrogenous base attached to it. Now, so this is the structure of a nucleotide, but this is one nucleotide. This hydroxide will again form a bond like this with the next nucleotide, CH2, and there is CO, C, and this is the nitrogenous base. Like this, I mean like sorry, I'm so sorry. This hydroxide will form bond with the carbon atom 3 of the next nucleotide. Let's see the bond forming. We know that this carbon have hydroxide, hydrogen I showed you. These two will form another ester bond. Let's see. Okay, let's draw another nucleotide here. I'm doing this so that you can imagine. I only wish that the board was a bit bigger. There is carbon, there is carbon, and this is the base, nitrogenous base, and there is CH2, blah blah blah. There is the hydroxide, there is the hydrogen. Once again, water will be removed, and thus this carbon atom will form a bond. Like this, so on, the polymer formed. So from this phosphate ion, which was the phosphoric acid, became the trihydrogen phosphate ion. There are two hydroxides. One hydroxide will form ester bond with this carbon number 5 of one nucleotide, and the other hydroxide will form ester bond with the carbon number 3 of the next nucleotide. So they are called 5 prime 3 prime bond. Okay, this was an explanation. Now let's see from the book. Okay, so the pentose sugar can be either ribose or deoxyribose, as the name suggests. Deoxyribose is almost the same as ribose, except it has one fewer oxygen atom in its molecule. Figure 6.2 shows 5 different nucleotides from which DNA and RNA molecule can build up. Figure 6.3 shows the structure of their components in more details. You don't need to remember the structure, but if you enjoy biochemistry, you may find them interesting. Do not confuse adenine with adenosine, which is part of the name adenosine triphosphate. Adenosine is adenine with sugar attached to it. And don't confuse thymine with thiamine, which is a vitamin. Okay? So there is the structure of ATP. You need to learn this. Everything from the book you need to learn. Although ATP is not part of DNA and RNA, we'll look at its structure here because it's very similar to a nucleotide. How? We have a pentose sugar, we have a nitrogenous base, but we have 3 phosphate ion around here. So its structure is shown in figure 6.4. Adenosine can be combined with 1, 2 or 3 phosphate group to give in turn adenosine monophosphate, adenosine diphosphate and adenosine triphosphate. If it is joined to 1 phosphate, then adenosine monophosphate. If it is joined to 2 phosphates, then it is adenosine diphosphate. If it is joined to 3 phosphate, then it is adenosine triphosphate. So adenosine is ribose and adenine. If there is other nitrogenous bases, then that wouldn't be adenosine. Okay, now polynucleotides. To form the polynucleotides, DNA and RNA, many nucleotides are linked together into a long chain. This takes place inside the nucleus during the interphase of the cell cycle. We'll learn about the interphase in, I think, reproduction chapter or gene chapter, but there is a chapter where we'll learn in detail about the interphase phase. But basically speaking, interphase is the phase where the DNA content, that is the number of nucleotides, polynucleotides, increase either for mitotic mutation or meiotic mutation. Sorry, mitotic division, cell division, or meiotic cell division. Figure 6.5a shows the structure of part of a polynucleotide strand. In both DNA and RNA, it is formed of alternating sugars and phosphate linked together with the bases projecting sideways. The covalent sugar phosphate bonds called phosphodiester because it is an ester and there is a phosphate group. It is an ester bond. There is a phosphate group involved, and 2 ester bonds are formed. One with the oxygen of the carbon number 5 of a nucleotide, like pentose sugar, and another with the carbon number 3 of the pentose sugar. Okay. Link the 5 carbon of one sugar molecule and the 3 carbon of the next. The polynucleotide strand is said to have 3 prime, 5 prime ends. DNA molecules are made of 2 polynucleotide strands lying side by side. Running in opposite direction, the strands are said to be antiparallel. The 2 strands are held together by hydrogen bonds between the bases. The way 2 strands line up is very precise. Okay. So, we have seen an individual nucleotide. Now, let's go a bit in more detail. So, phosphate, and so on. And there we have the nitrogenous base. If this polymer forms, then this is a single strand. But DNA contains double strand. But RNA contains a single strand. So since DNA is double stranded, there should be 2 strands. And the way they are like joined together, it is very precise. If one strand goes in this direction, then the other strand will form in the opposite direction. When two strands are formed in opposite direction, they are said to be in anti-parallel. Okay, there is the nitrogenous base. Again, there is the nitrogenous base. It will be easier if I showed you this upside down. Never mind, no upside downs. And there is the nitrogenous base. Now, we have two strands anti-parallel, but they need to be joined by a bond. And that bond is hydrogen bond between the bases. This base should be here. So the two strands are anti-parallel, joined together by hydrogen bonds. Now, the number of hydrogen bonds formed are very precise. That's because the hydrogen bond between and the bases are complementary. That is, not any bases cannot join in any base. Adenine cannot join with thymine, guanine, cytosine. It's not upon their wish. Adenine, guanine, cytosine, thymine. Adenine can only join with either thymine, or if it's an RNA, then uracil. And guanine can only form bond with cytosine. Even the number of hydrogen bonds are specific. Adenine forms 2 hydrogen bonds with thymine and uracil, and guanine forms 3 hydrogen bonds with cytosine. So you may have one question. I just told you that RNA is single stranded. So how is there hydrogen bonds forming? Yes, that is a question, and you will understand in protein synthesis. But right now, just trust me and learn by heart that adenine with thymine forms 2 hydrogen bonds, and guanine with cytosine forms 3 hydrogen bonds. And instead of thymine, if there is uracil, then it will also form 2 hydrogen bonds with adenine. But adenine cannot bond with guanine, or guanine cannot bond with thymine. Neither cytosine with adenine. Okay? So they are called complementary base because one needs to be bonded with another as a complement, and they are fixed. So thus, we have known that adenine guanine is purine, while cytosine, thymine, and uracil is pyrimidine. Purine can form hydrogen bond with pyrimidines. Okay, let's see here. The bases can be purines or pyrimidines. From figure 6.2, you will see that the two purine bases, adenine and guanine, are larger molecules than the two purines, cytosine and thymine. So this is another point. In a DNA molecule, there is just enough room between two sugar phosphate backbone for one purine and one pyrimidine molecule. So purine in one strand must always be opposite a pyrimidine in the other. In fact, the pairing of the base is even more precise than this. Adenine always pairs with thymine, while cytosine always pairs with guanine. Adenine, thymine, guanine, and cytosine. A with T, C with G. This complementary base pairing is a very important feature of polynucleotides, as you will see later. DNA is often described as a double helix. So DNA is a double helical structure. Since it has two strands, DNA is made up of two strands. The strands are not like just straight. If one strand is like this, the other strand will be like this. And there are the hydrogen bonds. These are the hydrogen bonds. So this and this are the polynucleotide chains. Nucleotide chains. And these are the hydrogen bonds. So now you know why were there this structure. They are in a helical structure. That is, if you look in a 3D shape, it will be like this. And the other strand will be like that. If you search the internet, you can see them in an animated version. I can only show you the 2D version. To watch the 3D version, you need to search the internet. This refers to the three-dimensional shape that DNA molecules form. The hydrogen bonds linking the base and therefore holding the two strands together can be broken relatively easily. This happens during DNA replication and also during protein synthesis. We will see them in protein synthesis chapter and DNA replication later. And also, as we will see, the breaking of the hydrogen bonds is a very important feature of the DNA molecule that enables it to perform its role in the cell. RNA molecules, unlike DNA, remain as a single strand of polynucleotide and can form very different three-dimensional structure. We will look at this later in the chapter when we consider protein synthesis. So, the DNA has a precise three-dimensional structure, that is a helical structure, but the RNA do not have a precise structure and it is rather complex. It could be like, this is the single strand and the RNA molecule is like this. It could be. We can observe it using some ways, X-rays, diffraction, and there are other chemical ways, chemistry ways by which we can know the structure.